off you go.
Yeah, brilliant. So yeah, thanks everyone for coming. Hopefully, yeah, you'll enjoy my talk, and hopefully you'll have some idea already about what it's going to be about. But if you don't, then yeah, my name's Abigail Lowe and I'm Science Officer at the National Botanic Garden of Wales. But for the last 4 years, I've been doing my PhD based at the garden. So in partnership with Bangor University, and the kind of premise of my PhD was looking at pollinator foraging, specifically using a process called DNA metabarcoding, which I'll obviously explain, to identify what plants pollinators are using. So you're probably all quite aware that pollinators and insects in general are really important parts of the ecosystems. So not only do they contribute to ecosystem services, they're also just, so ecosystem services are things like pollination. They provide, that by them pollinating plants, they provide food to other animals, but also to us. And they're just, they're such vital components of the ecosystem that without them, it wouldn't be great. But unfortunately, many species are in decline, and it's actually estimated that 1/3 of wild pollinator species in the UK have declined between 1980 and 2013. So wild pollinators are things like hoverflies, bumblebees, solitary bees, moths, butterflies. And a lot of these declines are due to things like habitat loss, increased pest and disease, climate change, and also pesticides, increased use of pesticides. So they're really important, but they're in decline. So we need to conserve them, but how can we conserve them? So as I said, pollinators are a really important part of the ecosystem, and we know quite a lot about their ecology, but also there's a lot of stuff that we don't know. So we don't know too much about what plants they use, and something that is kind of really important is that if we know exactly what plants they are visiting and that they are feeding on, then we can help to provide these in gardens and in the wider landscape, and kind of it will help us to realise what habitats are important. And actually, gardens are really important habitats. So kind of statistic that I heard a few years ago now, which I've kind of, ever since I've heard it, I've not really stopped thinking about it and telling people about it, is that the area of gardens in the UK is actually greater than the area of national nature reserves combined. And like when you think about that, it makes you think, well, we put all of this, all of this importance onto nature reserves and, the wider landscape and protecting special habitats, but actually, not a lot of people do put that much effort into, looking after their garden and making sure that their garden is like a haven for insects. So, yeah, this kind of, and I'm sure I'm probably preaching to the choir here, because I imagine that you guys being part of Carmarthen Meadows group or being interested in this talk are probably, interested in wildlife and do wildlife gardening. But, we need to stop this nonsense of astroturfing lawns and concreting drives and things like that. So yeah, a lot of my PhD is about kind of finding out exactly what plants are being used, and then how we can use this information to kind of talk to the public and provide these like good habitats so we can help stop decline. So you often see lots of kind of plants for pollinators lists. So if you just typed into Google, what are the best plants for bees, or what can I plant for pollinators, you'll find loads of information, but there's actually quite a lot of issues with this information. So one of the things is that the lists between them are quite inconsistent. So you know, they're listing whole hosts of plants, but you know, what one place is saying plant these plants and the other place is saying, you know, plant these, and it's not really very clear where this evidence is coming from. A lot of it is anecdotal evidence. You know, some of it is, there's sometimes a bit of scientific evidence behind it, but a lot of the time it's anecdotal. Not only that, but there's other issues with things like, you know, going to a garden centre and picking up a plant with a perfect for pollinators label on it, and the plant has actually been, I don't know, imported from the continent, sprayed with pesticides, grown in peat. So yeah, there's so much work that we can do to just improve the habitat that we're providing, really. So a lot of the work that we do at the garden is kind of about just improving people's like access to good information. So one of the ways that you can figure out what plants pollinators use is to look at the pollen on their bodies. So you know, as you probably know, the pollinators go between different plants, feeding on pollen and nectar. And when they do this, they pick up lots of pollen. So some insects like bumblebees and honeybees, they will collect pollen and they'll mix it with a bit of nectar and they'll pack it on their legs, and then they'll take it back to the colony then. Some insects like solitary bees, they'll also collect the pollen, but they don't make it wet, so it's a bit you know, drier and it comes off a bit more easily. So that's why they think that solitary bees might be actually better pollinators than some bumblebees and honeybees, because the pollen is getting transferred off them quite easily when they go between flower to flower. And so yeah, so when they're moving between all of these different plants, they're picking up pollen. And so you can take the pollen off their bodies and you can identify what plant this pollen has come from. And so kind of traditionally this had been done using microscopy, so literally looking at pollen grains down a microscope. But this is really you know, the people that are able to do this are quite few and far between. You know, these days it's quite a skilled you know, it's quite a skilled person that's able to do this, and also it's really time consuming, and there's a lot of these plants which the pollens within the same family quite hard to identify. So I think things like grasses, the pollen of grasses they look really, really similar. So if you're looking at it underneath the microscope, it's actually quite hard to tell which species of grass it is. So instead we can use DNA. So DNA is quite nice because it can help us to, you know, and yes, there's obviously skill involved in the molecular techniques, but it's a lot easier to kind of train people in those sort of skills. And we can get pollen from, as I said, from the bodies of insects, just as in the kind of, so you can see here, I don't know if you can see my cursor at all, but the photo here is Aerostylus Intrakaria. So it's a hoverfly, but it looks like a bee. So it's quite furry, and you can just see like tiny little dots all over its body, and that's pollen. So hoverflies don't collect pollen like bees, but they will get it kind of all over their body. So we can get it that way. We can get it by scraping pollen off of legs, or we can sample honey, and there's pollen left in honey as well. So a little bit about kind of how we identify the pollen. So we use a process called DNA barcoding, and kind of without going into like way too much detail, basically a DNA barcode is a small section of DNA which has got enough variation between species so that it can discriminate between them, but not a lot of variation within a species. So it's kind of like the kind of analogy that I always try and use is that we get this, that we get this sequence, we sequence the DNA, and what we've got to do then is we sequence this very small section of DNA, and then we compare it to known sequences of plants that we know the DNA sequences of, and that's how we identify them. But in order for us to be able to do that, we of course have to have that reference library available of all of the known sequences of plants. And so Wales was actually the first nation in the world to DNA barcode all of its native flora. So we've been banging on about this now for, 10 years, which is a really long time, but I think it's quite cool. So it was, just over 1,000 plants that we went out, collected specimens of plants, got them identified by trained taxonomists, and sequenced the DNA of those plants, and then created a reference library. So we knew exactly, right, this is Rubus fruticosus Bramble, this is its DNA sequence, so that when you then sequence some DNA from pollen or honey and you've got an unknown sequence, you can compare that back to what you've got. And last year we actually finally got it published. The work was complete a few years ago, but we finally finished all the analysis and got it published to finish the rest of the UK. So now it means we've got this really nice reference library of DNA sequences that we can use. So as I said, there are more than just one type of bee. There's more than just bees are pollinators, but my research, most of the research that we do at the Botanic Garden is focusing on hoverflies and bees, specifically honeybees, bumblebees, and solitary bees. But I made this pie chart really just to kind of visualize really like the number of species that we have of each group. So a lot of people know about honeybees, but there's only one species. A lot of people know about bumblebees, but we've only got 24 species in the UK. We've got over 250 species of solitary bee. So these can nest in the ground, in your bee boxes, bee hotels, in trees. They've got such diverse life cycles and ecology. And then we've got even more species of hoverflies. So hoverflies, they, as the name suggests, they are flies, but in America, they actually call them flower flies. So that kind of explains in the name. They visit flowers and they eat pollen and nectar like bees do. So they're really important pollinators. So the garden is like a really good study site actually for looking at what plants pollinators use, because hopefully most of you will have been, if not, be aware of how the garden is set up. But it's, as well as being a formal garden with lots of horticultural plants, we're also surrounded by National Nature Reserve. So we're over 500 acres, and we've got over 5,000 plants from across the world. So in this site, pollinators really do have a choice of choosing native or non-native plants. And we can go out and sample them and find out exactly, what do they prefer. I just put this slide in just as a bit of interest, because it's something that I've been interested in, which has not kind of directly been part of my PhD, but I've been recording species at the garden by, just as you do, like biological recording, and I've recorded 68 bee species and 57 hoverfly species, which sounds like a lot, but it's actually a lot lower than I would like it to be. And I'm going to improve that number this year. Now that I've finished my PhD. But some of the notable ones are Longicornis. So that's a Section 7 species, and wasn't seen in the county since 2005, before I recorded it in 2019. We actually saw it again, a male and a female this time in the summer in 2021, which I was really excited about. The long fringe mining bee, Andrena congruens, that hadn't been seen in the county since 1985, and Sphacodes scabricolis, which hadn't been seen since 1997. So that just goes to show really that it's probably a bit of both. Like, the garden is a really amazing site. It's really diverse. It's got so many amazing habitats, but also Carmarthenshire is pretty under-recorded, probably with lots of species, but particularly for bees and hoverflies. I find that I find things which haven't been recorded much, but they should be quite common. So yeah, if you are interested in hoverflies and bees and things, and you want to, or you do any recording, yeah, just get in touch with me and things, because I'm trying to improve that a little bit better in Carmarthenshire. So what I did was I went out every month between March and September and collected any bees and hoverflies that I saw along a set of transects across the site. And I caught 382 insects. I then washed the pollen off them and extracted the DNA, sequenced the DNA, so that I could compare it to our reference library and identify what plants we used. Now, just a kind of quick overview of the results. So over those 382 bees and hoverflies throughout the year, over 2 years, I identified that they were using 191 different plants. Now, some of these were identified family level, some to the majority to genus level, but some to species. But actually only 6 of these plants were found on over 50% of samples. So these are the plants which are kind of principal importance, really. So on the right, I've just got a kind of illustration of the 6. So although there's 9, I will explain. So the most common plant which occurred on samples was Rubus fruticosus, so blackberry. This is now becoming very, very clear in all the research that we do that this is probably the top plant for pollinators in the summer. They just absolutely love it. They're all over it. Might be because it's really, really abundant. It's also a really good nectar source, so it's probably a bit of both. Then the second one was Brunculus and Vicaria. So these are actually grouped together because Vicaria actually used to be within Ranunculus, so that explains why the DNA is not able to split them very well, but we know that they have quite different flowering times. So basically we know that in the spring, if they were using the Ranunculus / Vicaria group, then that was the celandine, and then more towards your kind of May time, that's the more of the buttercups. Then the third kind of most abundant species was this group of 3 things which annoyingly are very, very hard to distinguish using DNA, but also grow in pretty much exactly the same habitat at the same time. So you can't even use that as a distinguisher. So we unfortunately are left with this group of thistles, knapweeds and catsia. And the thistles and the knapweeds could be there could be some horticultural taxa being used within that. We don't know, we can't distinguish between them, but given the abundance of lots of different thistles and the knapweed, like in our meadows, that it probably is the native species. And then Cassia is pretty distinct. There's not really anything else that could be confused with that. Then Angelica and hogweed, they are grouped together, so that was really abundant on samples. Again, they flower at pretty much the same time, but you know, it's pretty strong that it was these kind of two genera, well, species actually, because although you can't distinguish them, there's no other species that they can be. And meadowsweet as well, so Filipendula. And then, so that's 1, 2, 3, 4, 5. The sixth one was Asteraceae. So we had a lot of sequences which were just attributed to the daisy family. So we can't tell exactly what daisy family they were, but there's a lot of different things it could be. It's also the biggest family. I think the biggest family. You guys will know that more than me. Either the biggest or the second biggest. So then I looked at what were the differences between the groups. So are hoverflies and bees foraging similarly? And this graph, you know, you don't need to like study it loads to, you know, to work it out, but I'll just kind of point out the key thing really is that when I looked at the proportion of sequences for the Diptera, so that's your hoverflies, that's the taxonomic order that hoverflies are within, and Hymenoptera, so that's the bumblebees, honeybees, and the solitary bees together. The key differences here that you can see are that there is the bees don't use Angelica and hogweed as much as the hoverflies do. They do use it, but it's in this big bar here of taxa below 5%. So they use it, but it's not a major resource for them. The hoverflies use less of the thistles. They use loads more of the Brunculus and Vicaria and less of rubus. And we think that this is mainly because hoverflies, the way that they forage is they have these tongues which work kind of like a suction pad, and so they tend to like plants which are kind of open, flowers which are open, and their nectar and pollen are kind of more accessible than something like a thistle, where although the flowers look open, you know, you've got these really long corollas and the bees have got to have long tongues to kind of reach in. So we think that's why, but it shows quite nicely that they do forage quite differently. I then looked at, as I said, whether native or non-native plants are preferred. And I looked at this like throughout the year. And so you can see here, I looked at the proportion of DNA sequences which were attributed to native and near-native, naturalized or horticultural plants. Now the near-native category, that comprises things like, so I said a lot of the identifications were to genus level. That kind of obviously opens up some difficulties then when you're trying to classify something as native or non-native, because you get things like Prunus, which, yes, we've got, you know, native Prunuses, but we've also got horticultural Prunuses. So do you call Prunus native or non-native? And actually Prunus as a genus, the ones that we have available in the garden anyway, the majority of those are ecologically similar. They, you know, they've got similar ecology. There's no, apart from obviously if you have anything double-headed, then that would be a reason that a pollinator wouldn't visit it as a horticultural variety. But if you have a native and a horticultural variety of plant, and yes, there could be things like it has reduced nectar in the horticultural versions, but as far as we are aware, the horticultural and the native Prunuses are equally as you know, beneficial to pollinators. So we grouped them as near-native. And what we can see is that throughout the spring, most of the plants used are native. Same for summer, but there is an increased amount of naturalized and horticultural species used, and then by the autumn, most plants are horticultural and naturalized. So this kind of shows that towards the end of the year, that horticultural plants are quite important. So particularly clematis, heuchera, bidens, coreopsis, and Rudbeckian helenium. And this big chunk of naturalized plants, that's all Himalayan balsam. So we know that Himalayan balsam is unfortunately really important late nectar source for pollinators. I put this in really last minute, and this is again just a bit of a confusing graph, but what I wanted to show you really was each of these is the network of all of the insects and all of the plants. So it shows it's really, really complex. This is what it looks like in spring. You've got loads of your buttercups and your celandine being used. Then in the summer, it's different colours, so different plants being used. You've got your thistles here, your bramble, meadowsweet, lotus, and then by the autumn, totally different plants again. So you've still got your rubus, Himalayan balsam, clematis, bidens. So it just shows really how that there is such a change in foraging throughout the year. And then, yeah, so this slide really then is just to say that the main thing that we can kind of like take home from our work is that the plants of pollinators lists need to be, they need to specify between seasons. So you need to say in the spring, plant these plants for bumblebees, plant these for honeybees or solitary bees, because each group had different preferences throughout the year. So yeah, I won't spend too long on that actually. So yeah, actually that was the bulk of my research. So I'll move on now to a bit about my colleagues' work. So it's all like really closely related. So my colleague Lucy, she was looking at the effectiveness of commercially available seed mixes to attract pollinators. So our seed mixes, which are marketed as good for bumblebees or good for pollinators, are they attracting pollinators any more than a mix which isn't designed for pollinators? So if you visited the garden in 2018 or 2019 in the summer, you'll have seen Lucy's amazing plots by the bandstand, which she spent hours and hours looking after and sorting out. And she was using the same techniques again, so sequencing the DNA of the pollen on the bodies of the insects to identify what plants they were using, but particularly to see like what plants in the mixes are the pollinators using, and also how do different seed mixes compare. So she had two mixes which were designed for pollinators. So that was a bumblebee mix and a general pollinator mix, and then two which weren't designed to attract pollinators. And so this is just one of Lucy's graphs from her, oh sorry, I don't think I said actually, sorry. So Lucy handed her PhD in October. So there'll be even more, we're hoping this year that there'll be lots of papers coming out from us. So hopefully all of this research will be out there really soon. But just to kind of give an overview really of the number of individuals that she found in each mix and the number of species really. So each colour line relates to a different mix, and you've got from June to October for the three groups. So pretty much for bumblebees, the seed mix which did the best at attracting the greatest number of individual bumblebees was actually the commercial bumblebee mix, which is quite nice. For the hoverflies, it was the native mix. For the, I just have to move my video. For the solitary bees, there actually wasn't, she didn't sample many solitary bees, so it was too difficult to say much about solitary bees. Then when she looked at the number of species which were attracted, for bumblebees, there's only about 7 species that are common anyway, so you probably wouldn't see, and I think Lucy only had 4 or 5 species in her plot, so you wouldn't see a huge variation there anyway, which explains this graph. Hoverflies are much more diverse, and she found that the mix which attracted the most hoverflies throughout the year was the commercial pollinator mix. And then again, solitary bees, probably not enough data to say. And then Lucy looked at, again, what proportion of plants in the seed mix were being used. So not, what plants are being visited at any one time, but what proportion of species which are actually in the seed mix are being used. Because you'd think if a seed mix has been marketed as, this is the best mix of species that we can give to pollinators, then you would hope that you would have like a really high proportion of those species being visited. And whilst they might not all flower at the same time, this was the proportion of ones which were visited which were in flower. So you can see just by this graph really, so each colour is again a different seed mix, and there was never less than about 54% of species were being used in July in the aesthetic seed mix. So if we look at one of the seed mixes which has been specifically targeted for bumblebees, actually maximum amount of species which were ever used were 35%. And this is across all pollinators, but if you look at the more broad pollinator mix, again 50%, but in June only 40%, and in October 30%. So there definitely can be improvements on what plants are being used in these seed mixes. So Lucy did actually do some further work on designing her own seed mix, and we are really hopeful that at some point we will be able to market that as a thing, because just there's so much potential for these things to be improved, knowing what we know. So hopefully that'll be something that we can work towards in the future. So Lucy also looked at, so similar to me, so she looked at what plants are bees using, the bumblebees, and what plants are the hoverflies using. And again, the nice thing about all of this research is all of our findings are, we all agree with each other when we're doing separate projects, which is always nice, finding something crazy. That Lucy found that throughout the season, the greatest proportion of DNA sequences were attributed to this purple color here, which is the thistle Centaurea group, so knapweeds, thistles, which is the same as what I found in a totally different system. And then for her hoverflies, they didn't use the thistles as much. They used things like Coreopsis, Cosmos, Anthemidae, which is your chamomile tribe, Asteraceae, Glebionus sigitum. So again, the kind of more disc-shaped flowers. So moving on then to just lastly then, my colleague Laura Jones, so another science officer at the garden, who has her PhD was looking at honeybees, so how honeybees forage throughout the UK and also in the garden. And this research was also published this year. So Laura sampled honey from beekeepers across the UK and used DNA metabarcoding to identify the species in the honey. And so she sampled 441, she had 441 samples of honey from across the UK, which is really nice, analysis of what pollinators are doing, really, because it's all well and good us knowing what they're doing in the Botanic Garden, but is the Botanic Garden unique? Are they doing the same things elsewhere? And actually what we found, like, doing this work, was that the top plant, as I said, is Bramble. So Bramble was the taxa which was found most frequently across samples, followed by white clover, brassica, so your oilseed rape and relatives, hawthorn, apple and cotoneaster, sycamore, oak, rose. So there was kind of like maybe 10 or 11 plants which were really abundant across honey samples, and also really dominant in the honey samples. But then there were a lot of other plants which were present but at lower levels. And that's what we tend to find with honeybees, that although they are super generalist, that they do use a lot of plants, but because they forage for such a long time throughout the year, that it's inevitable that they are going to be generalists, but they're not as generalists as we think, in that they do use a lot of plants, but they use a small number of plants really frequently. And that's something which is becoming like really clear with our research. But the main kind of part of this publication really was a comparison then to, so this survey was done in 2017, but the last national honey survey was done in 1952. So in 1952, there was a man called A.S.C. Deans who took a survey of honey from beekeepers throughout the UK, and he identified the pollen within the honey. Now, obviously, he didn't use DNA to do that. He did it through microscopy. And actually, there was a really strong correlation in the plants which were found in 1952 and 2017, which showed the honeybees, their foraging has stayed quite similar in terms of the plants that they are using, which was quite nice, because that shows then the methods are quite comparable, although there were some things that weren't as easily identifiable in the microscopy, which we could identify to a higher power with the DNA. We matched everything at the same level to make it easier. But looking at the top 4 plants, they were the same in 1952 and 2017. So white clover, your hawthorn, apple group, with Prunus in there as well. So that's what I meant. We can split out Prunus with the DNA, but you can't with microscopy. Bramble and brassica. But the really cool thing that we could do was look at how the proportions had changed in the honey between over 65 years. So if you know, well, I think we all are very aware of the kind of changes that we've had in the landscape since the 50s. So we've obviously had increased agricultural intensification, which has led to, the plants have totally changed, and you'll see things like reduced clover lays, and that's really obvious then in the survey that we've done, is that the percentage of samples that had white clover in them was much higher in 1952 than it was in 2017. And also was the proportion of samples where this was dominant, so that's like the red. And when you relate that to changes in the countryside survey, white clover had a 13% reduction. So it just transcribes that, really. So it says that there was less clover in the landscape, so the honeybees foraged on it less. And similarly with the hawthorn group, actually, no, sorry, take that back. There was no change in the landscape. No, that's not right. That is too difficult to establish because of the grouping, but you can see that less have been used, and probably will say that that's because there are less hedgerows. And no change, you can't really see a change with the heather, but we have lost it in the landscape. Reduced red clover, massive increase in bramble, corresponding with an increase in the availability in the landscape, increase in sycamores, increase in brassica, so that shows increase with because it's been used as a crop, and then a huge increase as well in the Himalayan balsam, which became invasive in the 80s, I think. So yeah, this is really nice that we can actually use honeybees as kind of like a monitoring tool, really, to show that their foraging reflects what's happening in the landscape. So that was really nice. So just to kind of like finish up on a few things, whilst it's really important to provide plants for pollinators in your garden, it's also just as important to provide nesting habitat. So you can do this by providing bee hotels or some bare ground, or you can make a bee bank, and there's lots of information about this online. So I wrote a blog it's probably about 4 years ago now, actually, on bee hotels that you can find on our website, with a few different designs of ones that you can do, some like really easy ones like the plastic one here, which I think people do have mixed views on, but I find that they're a really good engagement tool, especially with children. And then there's also so on Bug Life's website, they've got lots of information about how you can make bee banks and things like that. And then just a quick plug, really, to say about the Grow in the Future project, which if you'd have gone to Kevin McGinn's talk, which was in October, I think, he probably would have talked a bit about this. So Grow in the Future funds me currently, and it's a 5-year project, which is coming, it's in its last year now with the garden. And it's all been about how we can use gardens and gardening to kind of improve health and well-being for people. We've been running courses and things like that. So it's been a really successful project, and hopefully, we'll be able to continue it somehow if we can get some funding, which is, we're trying. And then again, I don't want to kind of overlap too much on what Kevin may have said previously, but just, if you're not aware, or if you've just come to this talk alone, is that what I was saying at the beginning about all these problems with the lists and these stickers. We've actually developed a Saving Pollinators assurance scheme at the garden now, which means that we've got these stickers that we can put on plants, and we're working with 29 nurseries in Wales on this scheme, where they have grown these plants. They're peat-free, they're pesticide-free. We can trace exactly where they've come from, and they're plants which have been proven to be used by pollinators by our research. So this has been really, really cool thing to be part of, to be honest, because as a researcher and as an academic, you don't really tend to see your work have like a real, I guess so, like a real world impact like straight away. Sometimes it can take a while, and sometimes it's quite hard to like get the message across to people, particularly in kind of like what we do anyway. And I think this is like a really nice way that we can interact with consumers and things. So there's loads of information about that on the website. You can find out which growers you can buy from and things, so to get your garden more pollinator friendly. And then another plug to say that we were harvesting our meadow seed, and hopefully, I think we're doing it again this year. So we've got individual seed and also mixed seed. And we've been working with lots of different organisations and landowners and things, because I think we are only one of 2 suppliers of Welsh native meadow seed. I could be totally wrong and someone will probably tell me that. But I think that is obviously really valuable for us to have like a source of Welsh seed. So yeah, I think this has been a really good, and again, this has been funded by Grow in the Future, and we wouldn't have been able to do any of this kind of without them. So yeah, I think that is actually bang on 45 minutes. I think I did well there. But yeah, thank you guys for listening. Hopefully that wasn't too overwhelming with graphs. And yeah, I'm happy to take questions.
Well, thank you very much indeed, Abby. That was excellent. If you want to stop sharing screen now, cool. That would be good. And if has anyone got any questions in the chat? No, not yet. So if you can think of any, put them in the chat. And in the meantime, there's a couple of things I'd like to ask. What you found about bees and hoverflies preferring different plant groups, for many years, it's quite clear when you watch insects foraging that on umbellifers you get loads of hoverflies, but bees don't seem that interested in them. And there's, you know, a huge area of these flowers available, but bees don't seem to be that interested in them. And your data supports that, doesn't it?
Yeah, definitely. So that's something actually, yeah, I didn't really kind of touch on that. So the kind of the value of using DNA is that you can get loads more information, but also the really nice thing is just when it kind of it shows you something that you think to be true anyway. So as I said, yeah, there's lots of kind of a lot of anecdotal evidence to, you know, and I've seen it and I know exactly what you mean. Like in the summer, the umbellifers are just full of hoverflies, and you really don't, you see a few bumblebees, but not a lot at all. But actually, there's not that much like scientific literature that kind of like confirms that. Like there is, but it's not as obvious as you'd kind of think about it as you'd think. And I think it's just developing that evidence base really. But yeah, I do wonder, I think it's got to be a case of like the accessibility. And I think because the flowers of the umbellifers are quite small, I think a bee does, it's probably something to do as well with like how much energy they can get from it, how hard they have to work. They've got to go between each individual flower. So for a bee, it's actually quite a lot of work, whereas the hoverfly is probably not as difficult. But yeah, it is cool to just, yeah, as you said, just to confirm that.
Yeah, another thing that you were showing the comparison between Laura Jones's data with the DNA pollen sampling and the 1950s work by Mr. Deans. Is there any equivalent information from years back on what other pollinators other than honeybees were doing? Do we know anything about how what they have used to choose might be different, you know, now from how it was 60, 70 years ago?
Yeah, that's a good question. I think definitely not. I don't think there's anything kind of like as thorough as ASC Dean's was, but there is a lot of information out there about there was a lot of work. So particularly there was a researcher called Mary Percival who did a lot of work on identifying pollen from the legs of insects, and that was in the 60s, I think. But yeah, it would actually be something like quite interesting to like look back on how things have changed, but not as far. I can't think of anything off the top of my head as in a kind of like national sense that we could do a similar comparison. But there is something that you can do, which I think we have kind of looked into in the past, is you can use museum specimens. So you can get pollen off of museum specimens and, well, you could do microscopy or DNA and look at how it has changed over time. And I'm not aware of any studies which have used DNA to look at, I don't even know off the top of my head now whether there's been much work, like microscopy work on it in the UK, but I know that there was a study which came out last year now, 2021, from America, that used these museum specimens of the rusty patched bumblebee, which is an endangered bumblebee in America. And they used DNA to look at the pollen over 100 years. They used specimens across 100 years. And they found that the bee was using pretty much the same plants over 100 years, which is quite a cool thing for conservation then, to be able to say, well, it's not, you know, there's probably lots of reasons that it's in decline, but you can then say, well, it's probably not in decline because it's had to change what plants it's using, or that, you know, there's none of this rare plant that they used to love, like that's declined, so they are declining. So that's like, yeah, definitely there's so much potential for what you can do.
That's amazing that you can use DNA from 100-year-old pollen on a museum specimen bumblebee. That's incredible.
Yeah, it's pretty cool. I'd be a bit scared to try it, to be honest. I'd be too scared to damage the specimen. But yeah, it goes to show like how once they're dried, like it can be conserved for a long time.
So for your work, you say you recover some pollen from, you know, the various insects that you've collected. How much pollen do you need to do this? Can you just a few grains is enough, is it?
Yeah, I think so. That's one of the really difficult things of working with pollen is that it's microscopic and it's a massive like well, it can be a massive contaminant, so you've got to be so careful doing the work. But yeah, with the things like the bumblebees, they are absolutely covered in pollen, so you've got no problems there really. But that was something that we didn't really know, to be honest. We didn't know what the threshold was that you needed, but I think I collected 300 for that, the chapter that I kind of went over in the talk. I collected 382 specimens, and I had successful sequences from 369. So I only had, you know, there really weren't many that didn't work, and all of the ones that didn't work were tiny little solitary bees and a couple of hoverflies. So it wasn't really surprising, but even there were quite a few that I was surprised. So it goes to show that they really didn't have to have many. And I also didn't. So a lot of people, when they do this sort of work, they only will collect insects with visible pollen on them. So like a pollen load, whereas I collected males and females. And yeah, I got like results from like it was really successful. So it goes to show that it doesn't just need to be like a pollen load. It can be, they're absolutely covered in it. So
yeah. One more question I've got before we open it up a bit more. The comparison between the 52 survey and what Laura did in, what was it, 2017? Huge increase in the importance of bramble for forage. Why? Why are they more? Why are they using bramble more? Is it because something else that they might have been using is not there now? I mean, for example, white clover. Is that why? I mean, I can't imagine that there's vastly more bramble available than there was then.
Yeah, so actually, I think when we looked at the countryside survey, there was actually an increase in bramble. I think they're just,
really?
yeah, so there has been an increase in scrub, but I do think that that switch is probably definitely a factor. So yeah, they're looking for a replacement, and white clover and bramble are both high in nectar. Bramble's also got like really good quality pollen, so I think it's probably just a mix of both. But yeah, it has increased in the landscape, which kind of corresponds. I think those top four plants, so the white clover, the bramble, the sycamore, and the brassica, those changes were significant, so either an increase or a reduction, and they did correspond to the changes in the landscape, and some are more easily explained than others. Yeah, like the Himalayan balsam, that's really easily explained because there was none of it. Well, yeah, I'm not sure when it came, but there wouldn't have been barely any, if anything, in the 50s.
Yeah. Right, okay. There's a question here in the chat from Laura. As well as counting number of pollinator species, did you look at the biomass or frequency of bumbles versus hovers versus honeybees? Do you think the introduction of multiple honeybee hives would have a negative impact on resident pollinator species?
Yeah, so I think just trying to work out I think what you mean, yeah, by the first bit is so basically did I look at how abundant they were? I didn't, so I literally just went out and I just collected what I found, but I actually didn't collect as many honeybees as I did hoverflies and bumblebees. Lucy didn't she didn't look at honeybees at all, so that might have been, she might have found something there. So no, I didn't look at kind of the numbers in the landscape, but the hives thing is a tricky thing, and I don't know where I sit. I think I think in the whole the area there is like two like really strong sides to the argument, and whilst there is something to be said about too many hives, and there's also there's issues with irresponsible beekeeping, definitely that there is some of that, and the problem with the media and people thinking that having honeybees helps conservation when it's not that simple, but I think they are a really good kind of engagement tool, so they're like a really good way in. I think the main thing is, like, for me, the thing that I always come back to is firstly we totally need to understand like how these resources are being used by the different groups before we can start to understand if there is competition. And as long as there is plenty of resources available, then hopefully the competition is reduced. The biggest problem you have is in places like cities where there's no resources, so there's no green space, so there's not a lot of floral availability to begin with, and then there's hives on top of roofs, and you're right that then there would definitely be competition, but I really struggle to answer that question, to be honest, because they definitely forage completely differently, and they might outcompete them kind of in the short term, but I don't know. I hope that kind of doesn't go around the question too much.
No, I appreciate it. I completely agree with you that it is not a simple question. There are people, quite well-known people, who'll say, well, if you want to help the birds in your garden, you wouldn't get a load of chickens, as though that is the equivalent of having honeybee colonies, and it is not that simple. I mean, there is some evidence that honeybees may actually, in some cases, help other pollinating insects because they pollinate flowers that would otherwise not be pollinated. But it isn't. It's something that I think about a lot because I'm a beekeeper and a conservationist. So, but I've only got 3 hives, so I don't think I'm one of the really bad influences.
Yeah, it's something I'm always like, I'm Switzerland, eh? I just say
Yes,
it.
I agree.
Yeah, I think I was, oh yeah, that was just something I was going to say. Yeah, I think one of the main arguments as well is that they're non-native, whereas we don't. A lot of the evidence for the competition comes from places where they are introduced, and we actually, what is the definition of what is the definition of native? I think it's it's complex.
There is a subspecies of honeybee that is a northern European subspecies that is a wild bee. But we don't, because of all the diseases we've also imported, there are very few wild colonies left. But anyway.
But yeah, that's a whole other talk, isn't it?
Yeah,
like,
it is.
Hopefully that was somewhat helpful.
Okay, someone question here from Nigel. Do you have any thoughts with respect to winter flowering plants? We use hellebores, winter flowering honeysuckle. Any other suggestions?
Yeah, so that's actually a good question, because I didn't do any surveying through the winter. I mean the numbers that I found in the autumn were so low anyway that I think if I'd have tried that it wouldn't have been good. But from what I know, just from like observation and just what's in the literature, so hellebores are really good. Also, winter flowering honeysuckle, Mahonia is really good as well. So Mahonia flowers can flower like between December and January, and that is one of the ones where so there aren't any so it's only honeybees really which would be active throughout the winter on like a warm day, but you can get some buff-tailed bumblebees, so Bombus terrestris colonies, which you see workers in December and January, and that is mainly kind of in warmer areas, so like southern England, but they tend to use Mahonia mostly. But I actually saw a tweet just before this talk that someone saw a queen buff-tailed bumblebee in Baglan today, so that was really cool because it's the 6th of January. So
that's amazing.
It's cool, but then you're also like, is that global warming? Don't know.
Well, I suppose
really
another factor is, though, there aren't many native plants that would be flowering in the middle of the winter, so there would be nothing for them to forage on if it weren't for some non-native horticultural plants.
Yeah, definitely. And bumblebees are cold climate, like they are adapted to colder climates anyway. So yeah, I'm trying to think now to actually answer the question of winter flowering plants. We do actually have a blog on our website about we've got multiple blogs all about the best plants for the different seasons, because I'm thinking the snowdrops are a good one, crocuses. I'm trying to think of, this is the thing, I just forget everything exists in the winter, and then I'm like, actually no, there are things that we can do. But I have written blogs, so I should know off the top of my head. But if you go to our website and then probably science, grow in the future, there's like a section which is like for gardeners, or like resources for gardeners, and there's loads of stuff in there, and that is where hopefully there is a blog about winter flowering plants. If not, then I'm going to add that to my list of things to do next week.
Good idea. Yeah. Everybody, feel free to unmute yourselves now and join in if you would like. Anyone got any other questions?
I've actually just seen in the talk there is another question. Just how did I get into this area of research?
Yes, I was just going to come to that one, so please do.
by chance, I guess, which is how everyone always answers questions about their career, I guess. So without, God, I'm going to end up telling my life story now, but
It's all right, we've got time.
Yeah, I grew up about 10 minutes away from the garden. So I've been going to the garden ever since it opened. My grandparents are members. And I did some work experience. I think I liked biology in school, like, and then I did some work experience for a week when I was in school, and then decided to do biology at uni. And then through kind of my work experience at the garden, I then got on to do a placement year. So like a sandwich year between my second and third year of uni, where I worked at the garden for a year. And even at that point, it was very much, I was interested in plant genetics. So I didn't have a clue about insects. I didn't know anything about them really. It was just, I came into it from the kind of plant side of things. And on that placement year, that was when we did the pilot study for what turned into Laura's PhD. So we did the honey sampling and we sequenced the DNA to identify the plants which were in the honey. So that's when I kind of started to, and even then we had, so one of my colleagues, Andrew Lucas, he did a similar PhD on hoverflies. So I used DNA metabarcoding to identify pollen on hoverflies, and that it was only then really that I was like, oh, actually, what that is, I didn't know what hoverflies were. And then I did that year in industry. Was interested in insects, but wasn't like, oh wow, I found my calling, and then I don't think it ever happens like that, is it? No. And then went back to uni, and then thought about doing a PhD, and then me and Natasha kind of, we put in a funding bid, and that was it really. And then I started doing this PhD, and now I'm literally obsessed. So now it's bizarre because now I can't imagine doing anything different. But by total chance, I always say I've slipped through many a net, and now here I am getting things into a net. But it was just a bit of an interest in plants, which then spurred into, oh well, actually, I can't say what I was going to say because I'm talking to Carmarthen Meadows group, but I find insects more interesting than plants. But there's room for everyone, right? So
of course,
exactly. But also, to be fair, sometimes I'm like, I should study plants because at least they stay still and you can go back to them and you know exactly where they are and when they're going to be there. It's a bit harder for insects.
That's true. Yes, they're easier to catch.
Yeah, exactly. Trying to catch a tiny solitary bee that you can't even see is up there. Or get into a tube without getting any pollen on your hands, on the tube, on the net. Yeah, not nice.
There's a question here from Katie. May we put links to your website onto ours? And Katie's from the Epping Forest Beekeepers.
Yeah. There's plenty of information and resources on the garden website. Feel free to put them out there. They're there to be shared. Yeah. Love for people to have access to them. Definitely.
I'm glad you said that because we do it all the time on our website.
Yeah, definitely. We need those clicks,
Yeah, absolutely. Right. Well, other than questions, there's a lot of people thanking you for your talk and saying how fascinating it was. So if there's no more questions, I think we can call it an evening then. So thank you, Abigail. Thank you very much indeed. It was a fascinating talk. The next one is in just over a month's time when we've got Layla Thornton from NRW who's going to be telling us about the history and conservation of floodplain meadows. And there's a project going on not far from here, actually, on the Bishop's Garden, Abigail, which includes restoration of a floodplain meadow. So in the Towie Valley. So yes, we'll be hearing about that on February the 10th. That will be. So thank you very much indeed, Abigail. Thanks for all your interesting insight into the work you've done. It was very, very good.
Oh,
and we'll be putting the recording of your presentation on our we've got a YouTube channel for the Meadows group, and it'll be on there, but it'd take me a couple of days to do that, but it'll be on there in a few days. Okay, so thank you very much for coming, everyone, and thank you, Abigail.
Thank you. Thanks, everyone.